Other questions? All right. So today we're going to be talking about lipids and membranes, and this is going to be part of a kind of coherent whole because what I really want to be talking to you about is how signals get from outside of the cell to the inside of the cell, signal transduction pathways. And in order to do that, we need to figure out what separates the outside from the inside, how proteins behave when they're sitting in those membranes, and how the signaling molecules interact with those membrane proteins. So, yeah, let's begin just by talking about lipids, then we'll get into how lipids form membranes, then we'll talk about membrane structure and some models of membrane structure, and then we'll, towards the end, hopefully be talking about membrane proteins, and then we'll really launch into that on Thursday. So the important thing really about cells is that they are compartments. You probably could not create a living system unless there was some boundary between the entity that is alive and its outside environment. And so we need to create these barriers between what defines the inside of a cell and the outside, and it needs to have a bunch of characteristics. It needs to be semi-permeable. You can't just have a complete wall. You need to be able to bring things in and out, and that needs to be subject to some kind of regulation. So there are tons of membranes that exist in the cell. The exterior cell membrane is perhaps the most important, but there's also additional compartments within the cell, such as the nucleus, such as the endoplasmic reticulum, lysosomes, etc., etc. And each of those need to be defined by membranes, the components, the constituents that really define the interior from the exterior and allow for segregation of these different regions and from the cell with its exterior. So let's figure out what the cell uses to create these membranes and how they behave. So what makes up a membrane? In particular, we'll see that predominantly it's composed of lipids. Membranes, proteins sit within the lipid membrane bilayer. We'll see that there are sterols that sit in there and influence the membrane properties, and that the lipids, as they sit in these membranes, can be subject to modifications such as glycosylation. And we're going to get into all of those details, but you'll see that the predominant thing right here, these little blobby groups that seem to have kind of like a head and a tail and form this bilayer. So what are those? Those are lipids. The definition of a lipid is a biomolecule that's soluble in organic solvents, but it's poorly soluble in water. So these are essentially largely hydrophobic species. And it's a kind of a catch-all term that can be applied to so many different kinds of molecules. We'll be talking a lot today about fats, oils. Olive oil is a lipid. Vitamins can be considered lipids. Many hormones such as estradiol and testosterone are also lipids. And most of the non-protein components of membranes such as cholesterol. But let's get to perhaps the most basic lipid, which is a fatty acid. And this is an amphipathic molecule. We've talked about amphipaths over and over again in this class, but you'll see that it has all the features of an amphipath because it has a hydrocarbon chain. This is going to constitute the hydrophobic group, and then it's got a carboxylic acid group at the head, and so this is the polar charged species. And we know that amphipathic molecules have a tendency to self-organize in some pretty interesting ways. We've seen that in particular with regard to protein structures, and we'll see how self-assembly of these molecules leads to the formation of membranes. So I showed you a alkane saturated fatty acid tail, and the other type of lipids include sites of unsaturation, such as this alkene right here. And you can see right away that there seems to be a pretty important difference in the way that the hydrocarbon chain is organized. This seems to be nice and straight, and this has got a kink in it. And that really influences how the lipids behave when they begin to self-assemble. In particular, saturated fatty acids, they can pack against each other really, really nicely, nice and snug, with lots and lots of van der Waals contacts that really help to organize what can be, under the proper circumstances, such as temperature, almost a crystalline environment, because that packing is so regular and well organized. Whereas if we mix together saturated and unsaturated fatty acids, okay, so each one of these alkene groups right here introduces a little kink in it. And now as we try to organize these things together, yeah, that regular packing, it's not really happening. So it's going to be a lot looser. There's going to be a lot more essentially void volume, if you will. It's just not as nice and crystalline here. So you can probably tell that if we mix together saturated and unsaturated fatty acids, as they begin to assemble, they'll have different characteristics. So these double bonds that are present in many fatty acids really influence their structure. And we need to talk about how to describe these types of molecules, nomenclature essentially. So one of the things that we need to figure out is how long a particular hydrocarbon chain is. That will be an important characteristic, but also where the sites of unsaturation are. So very often we'll talk about omega-3 fatty acids, and you've probably heard all about these omega-3s, omega-6s. They're so good for you. We'll talk a little bit later about why they might be good for you. But for now, let's just figure out where to put our site of unsaturation. We're going to call this terminal methyl group right here the omega carbon, right? So the beginning would be an alpha position, and the end, going from alpha to omega, that's the terminal position. And then we can count back towards the head group how many carbons it is before we initiate our site of unsaturation. 1, 2, 3. Here's our alkene group. So we call this an omega-3 fatty acid. There's our double bond. And you'll see that we've drawn this right here as a cis alkene. It's important to note that that's only one of 2 isomers that can be formed, and we'll talk about trans fatty acids a little bit later. All right, so here's a list of many of the typical occurring fatty acids that you will see in biological systems. And the important things that you need to be able to recognize is that the length of the hydrocarbon chain can vary. Here's one of the shorter ones. This is a 12-carbon skeleton. We call that dodecanoic acid for obvious reasons, but they can become a lot longer, and they go all the way up to this tetracosanoic acid, maybe a little bit longer than that, but these are the common ones, varying between, let's say, 12 and 24 carbon units. All right, other variations. So these are all saturated fatty acids, and then we can begin to put in sites of unsaturation. So here's one that I think about a lot, oleic acid, very common from olive oil. And this is an 18-carbon skeleton. It's got 1 site of unsaturation. So we would be able to draw that right here, and we would call that a cis-9 octodecanoic acid. In this case, we're going to be beginning at this position and marching down towards the omega position. So when we use this systematic name, we're going to call this 9-octodecanoic acid, but we also have this alternative nomenclature for unsaturated fatty acids, these omega-3 or omega-6s. So you should be comfortable being able to identify these fatty acids using either type of descriptor. So, extent of unsaturation, the oleic acid has one alkene, but we can have more. So here's linoleic acid. This has 18 carbons in a skeleton, 2 sites of unsaturation, cis, 9,12, octadecanoic acid. We can have 3. This is linolenic acid. We can have 4, arachidonic acid. Okay? So these are important variables when we're talking about fatty acids. Length of the carbon skeleton, number of sites of unsaturation, positions of unsaturation. And you'll see that the physical properties of these fatty acids change quite a bit as well. In particular, we're going to talk quite a bit about melting points, right, where these fatty acids undergo a transition from a solid to a liquid. And so some of these stearic acids, this is actually Greek for hard fat. These are the types of fats that are very common in animal fat, beef fat, tallow, rich in stearic acid. And so these have high melting points, and they're typically solids at room temperature. Whereas oleic acid, right, we know that's a liquid, we pour it on our salads, and that has a melting point below room temperature. So we need to think about what exactly is going on, why these different structures of these fatty acids give rise to these different physical properties. So here's our trend, right? As we go to longer and longer carbon skeletons of these fatty acids, the higher the melting temperature. And that makes sense. The longer the skeleton, the more number of carbons there are to engage in van der Waals interactions, the more energy that we'll get by packing them together, the more difficult it will be to melt them and turn them into a liquid. More unsaturated bonds also give rise to a lower melting temperature, and this explains why olive oil is a liquid. And this relates to this extent of irregularity and kinking when we introduce these sites of unsaturation with regard to cis alkenes, because they just can't pack together in a regular crystalline array. So it gives rise to lower melting temperatures. The important interactions, as I mentioned, are van der Waals packing with the larger surface area giving rise to more intermolecular interactions. All right, that was not good. Excuse me while I get this up and running again. So now we're going to talk about how fatty acids are going to be used as far as storage of energy. And so we'll begin to pack the individual fatty acids together as triacylglycerols. So we'll organize 3 fatty acid chains onto a glycerol molecule, and we will use that as a storage form for hydrocarbons. So here's what we're going to do. We're going to take this glycerol right here, and we're going to append upon it 3 fatty acids, and we're going to do that by incorporating them as ester linkages. So here's our typical triacylglycerol. This is the typical storage form of fatty acids. And these are really great ways for us to store energy. They're used to store energy both in plants and animals. So why would we store our hydrocarbon units in this form of these triacylglycerols? We talked before when we were talking about polysaccharides storage as glycogen, for example, in the form of polysaccharides. Why do animals and plants convert these into these fatty acids and store them this way? Any suggestions why we might do that? So what do you note about these structures here that's really distinct from, let's say, starch or glycogen? You know that hydrocarbons can be stored as starch granules. That's pretty common. We talked about that when we're talking about saccharides. What's distinct about these structures? So these groups here don't look anything like glycogen or polysaccharide. Why not? Yeah. Okay, so they're linear, that's true, whereas saccharides tend to be formed into repeating cyclic structures. So that's one aspect, but what about the chemical composition? Yeah. Right, so these are saturated hydrocarbons, and so these don't include polar groups. So you can tuck these away into fatty droplets, just as pure triacylglycerols, whereas typically for saccharides, polysaccharides, because they're polar, they want to be hydrated. So the energy density of fat tissue is much, much higher than you would find for something like glycogen, which needs to be stored both as the polysaccharide in the hydrated form. And so a lot of the density, a lot of the composition of that is going to be in water. The other aspect is because these are pure hydrocarbon chains, these are all present in a higher reduction state. And so when we go into a metabolic cycle and begin to break these things down and want to try to get ATP out of them, because they're in a higher reduction state, we can get more energy out of them per carbon unit. So these two reasons are probably why we store our hydrocarbons as fuel in this form as these triacylglycerols. More energy per unit volume, per unit mass. And so here's what the storage form of these look like. These are adipocytes, fat cells, and these are fat droplets sitting inside those cells. And animals use these for 3 reasons. First of all, to have them tucked away for energy production. We can mobilize these fats, turn them into ATP using the metabolic processes we talked about last week. Adipocytes also are used for heat production. This is the typical function of brown fat. So when we need to thermoregulate, we can do that. And then also for animals that live in cold environment, these sorts of adipose cells are useful for insulation. So this aspect of different lipid composition giving rise to different physical properties can be seen immediately by looking at the difference between olive oil, butter, and beef fat. And these contain different mixtures corresponding to these different features that we were just talking about with regard to whether or not they're short here or longer fatty acids, whether they're saturated or unsaturated fatty acids, and the unsaturated ones are here. So this is 75% unsaturated fatty acid, and you can see that's a liquid at room temperature, whereas this butter right here, 65% saturated fatty acid, that is solid at room temperature. And here's beef fat, or tallow. So why is this harder than butter if it's got more unsaturated fatty acids than the butter does? So it's obviously not just a matter of whether or not it's unsaturated. There's something else that's going on, we already mentioned. Why is beef fat really solid, whereas butter tends to be a lot softer and melt at lower temperatures? Yes. That's a good suggestion, that we're taking a look at differences between cis and trans alkenes. We'll talk about interconversion. That's not what's going on here. Yeah. Right. So the beef fat just has a larger fraction of these longer longer saturated fatty acids, these C16 and C18 saturated fatty acids. It's got a larger fraction of those. So those were those stearates that I showed you on that initial table. The longer the hydrocarbon chain, the better the packing between them, the stronger the van der Waals forces that exist between those individual fatty acid groups. Okay, so what fatty acids in particular are present in typical dairy foods, creams, butters? So these are the kinds of things you might see as far as saturated fatty acids. Here's palmitic acid, and then here's oleic acid. Here's our cis alkene present, and so this is going to be an omega 9 fatty acid. And then in butter, we also have some nice small fatty acids as well, like this caproic acid. This comes from the Greek name for goat, and so this has got a goaty aroma to it. And this butyric acid is a very short chain fatty acid, and this has got a kind of a funky smell to it as well. So I'm kind of interested in butters, and I'm really interested in what happens as you break down the triacylglycerols and begin liberating free fatty acids. And I'm particularly intrigued with an unusual butter that comes from Morocco that's fermented. And I thought that this was really peculiar. Why would you ferment a butter? And it turns out that this is really nice when you're in an environment like Morocco, because it allows you to store these things at room temperature for long periods of time without having to worry about degradation and spoilage. So what happens during the fermentation process is that enzymes and microorganisms such as lipase catalyze the hydrolysis of these ester linkages, and they begin to clip off these free fatty acids from the triacylglycerols. And so some of these are a lot of the same bacteria used in these fermentations that you might find in some of your more pungent cheeses. And as you begin to liberate things like caproic acids and butyric acids, you get a funky butter. And it's actually quite tasty if you like your funky cheeses. So this is a dish that I inflict upon my family from time to time. It's a Moroccan chicken that's cooked in this funky butter. And this is this butyric acid that you kind of have to get used to. It's an acquired taste. I have a sample here of Moroccan fermented butter that has these free fatty acids liberated off the triacylglycerols if you want to come and check that out. All right. There are a variety of different ways to conjugate fatty acids onto a backbone and use them within the cell. So far, we've talked about these triacylglycerols, and these are storage lipids. This is what we pack into our adipocytes. All right, so we take 3 fatty acids and we conjugate them onto glycerol, like I showed you before. But there's other types of lipids that are commonly used in the cell, not so much for storage, but actually these are the lipids that we're going to form our membranes from. The two major classes are phospholipids and glycolipids. And again, we're going to take our lipid groups, we're going to conjugate them onto a scaffold, and the differences are in what those scaffolds are composed of and what we're hanging off of them. We've got phospholipids here, which contain negatively charged phosphate groups, phosphoesters, and we also have glycolipids in which we're hanging off saccharide groups off of our skeleton. So the glycerol, I've shown you before, these glycerol phospholipids contain two fatty acids, and then these phosphoalcohol groups. We'll take a look at what this sphingosine unit is in a bit, and we'll hang off phosphatidylcholine. And then with regard to these sphingolipids, we'll take the same skeleton right here, we'll append one fatty acid, and we'll also incorporate mono or oligosaccharide onto that. And so these are the different types of lipids then that we're going to use to make our membranes. Okay, so let's begin with glycerol phospholipids, right? So we're going to take, once again, our glycerol unit as our scaffold, our backbone. We're going to begin hanging things off of it. Right away, for all of these glycerol phospholipids, we'll start with glycerol 3-phosphate. So here's our phosphate group. And then we can conjugate, once again, just as in the triacylglycerols, saturated fatty acids or unsaturated fatty acids as ester linkages onto the glycerol. And then we'll also append a variety of different head groups as substituents of this phosphate as well. So this is the common structure for all of these glycerol phospholipids. So what are we going to hang off this position right here? That will determine these different types of glycerol phospholipids. Right? Just the basic skeleton itself, we call that phosphatidic acid. And then we can hang off ethanolamine and position a positively charged amine group right there. That will give us phosphatidyl ethanolamine. Choline gives us a quaternary ammonium group. We call that phosphatidylcholine. So we can incorporate either positive charges, negative charges. We can incorporate something like a phosphonocetol. So these have negatively charged phosphate groups appended onto a saccharide unit, and then that is conjugated onto the phosphatidic acid. Okay, so all of these then can become constituents of cellular membranes, and different types of tissue are going to be rich in different types of these different glycerol phospholipids. This is one that's common in heart tissue. This is a cardiolipid, and on the backbone, we will incorporate phosphatidylglycerol. Here are our sphingolipids, right? So now we're not using a glycerol unit as our scaffold. We're using this sphingosine group, but you'll see that there are commonalities. This sphingosine group has this hydrocarbon tail already incorporated into the scaffold. And this is, in essence, similar to the hydrocarbon unit that would be present on a conjugated fatty acid. So this has already got one of these alkyl substituents already appended onto the sphingosine of the sphingolipid. A variety of different fatty acids can be conjugated onto the sphingolipid, but now notice here that we're not using an ester linkage, we're using an amide linkage to attach to C2 of the sphingolipid core. And then once again, different sphingolipids, head appended at this position right here.
Like Caribbean, Puerto Rican
choline shows up.
I had some interviews for now residency. That's surgery.
So if we attach this phosphocholine group at this position, we get something called sphingomyelin. We can attach either monosaccharides, disaccharides, oligosaccharides at these positions of the lipid. So there are similarities, as I pointed out, between the glycerophospholipids and the sphingolipids. Once you realize that this unit that's present on the sphingosine is essentially acting as a fatty acid hydrocarbon chain, then the similarities to the glycerophospholipids become really apparent. So here's our phosphatidylcholine, two fatty acid groups appended through ester linkages. Here's the overall appearance of that group. And here's our sphingomyelin, again, two hydrocarbon chains, fatty acid chains, appended here onto the sphingosine core. So very similar, and that's why they can both be used as lipid constituents of membranes. So I think we talked before about the important biochemistry of oligosaccharide conjugates. The variable oligosaccharide presentation on glycosphingolipids will determine your blood type group. So depending upon what saccharides are being presented on the exterior of your cells and your cell membranes, whether or not you have, for example, a GalNAc residue or a lactose residue present on these glycosphingolipids will determine your immune compatibility with regard to donating blood. These are showing up on the exterior of your red blood cells. All right, so other important constituents of membranes include this critical lipid right here. This is cholesterol. And right now you're looking at this and going, gee, that doesn't really look that much like some of the other fatty acids that we've been looking at that are clearly amphipathic with a terminal carboxylic acid group and a more or less linear hydrocarbon tail. Nevertheless, it really does have a lot of the same features. Cholesterol includes a chemical structure we'll see quite a bit of, this steroid. This has got 4 rings within the steroid nucleus. It does have an alkyl side chain appended upon that steroid core. And then on the A ring, it's got this individual hydroxy group. This is the essentially the polar unit on cholesterol, and this defines it as being an amphipathic molecule. This tiny hydroxyl group is sufficient to tell the cholesterol group how to organize itself, that it wants to orient that polar head on the exterior of a lipid membrane as the cholesterol is being inserted into the membrane. We will talk on Thursday about how signals are passed through the cell, from the exterior of the cell to the interior of the cell, and we'll talk a lot about what those signaling molecules look like. In some cases they're peptides, in other cases they're proteins, in other cases they're nucleotides like cyclic AMPs, and I just want to point out that lipids themselves can function as cellular messengers. So one of these is derived from arachidonic acid. This is one of the unsaturated fatty acids we talked about as being an important constituent. This is the way it might be arrayed on a membrane phospholipid. And one of the ways that signals can be passed from the exterior of the cell to the interior of the cell is that this arachidonic acid group can be cleaved off the scaffold to liberate the free fatty acid, which then can be converted to intracellular messengers such as prostaglandin or thromboxane. And it is these molecules, inhibition of the formation of these molecules from arachidonic acid, that is the mechanism of action for non-steroidal anti-inflammatory drugs, also known as our common analgesics, painkillers. So the steroid group is going to be showing up a lot, not only in the form of these lipid membrane components with cholesterol, but a bunch of different hormones that are derived from cholesterol. And in particular, these two sex-determining hormones, testosterone right here, and estradiol. You'll see that all of these steroidal molecules have in common a polar head group that's present on this A ring, and that there are 4 rings that form the steroid core. So even these very subtle modifications, the presence of a methyl group here, for example, variations in the nature of the aromaticity in the ring structures, these will determine which of these steroid hormones are going to interact with which receptors and signal the types of gene expression that will determine male from female. And we'll talk a lot more about the action of steroid hormone receptors when we talk about cellular signaling on Thursday. Okay, so a big part of our discussion of saccharides was describing how individual monosaccharides or oligosaccharides can be used to decorate protein structures, to form glycoproteins. And just as we saw in those cases, we can also decorate proteins with lipid structures. So these turn out to be quite important because depending upon whether or not we have a lipid group attached to a particular protein, can determine where in the cell these proteins end up. And not surprisingly, if we put on a protein a long hydrocarbon group, and these can be either unsaturated, like these farnesyl groups, or saturated, like these myristoyl groups, these hydrocarbon chains are going to allow the protein to be associated with the membrane. These essentially function as anchors and localize these protein structures onto the membrane, either on the intracellular side or the extracellular side. The lipids can be attached either onto a protein side chain at a nitrogen, a sulfur, or an oxygen. Depending upon the type of lipid group and the type of enzyme that's used for this conjugation, you'll find that a lipid group can be attached, for example, onto this serine side chain. S-lipidation would be accomplished by conjugation to form a thioester to a cysteine side chain. Okay? So, just as we talked before about how to characterize peptide structures, how to sequence them, we also talked about glycomics a bit and how to characterize oligosaccharide sequence and the saccharide composition. Also just want to let you know that there are a variety of different techniques that are used now to characterize the different constituents of biological membranes. And this turns out to be quite important when you're trying to identify, for example, a particular microbial species that might be colonizing an organism as a pathogen. You might want to be able to identify it quickly, and because different organisms have different constituents of their lipid membranes, this can be used to provide a type of a fingerprint. The techniques that are used are just to obtain whatever tissue you're interested in. You then homogenize that tissue. You do a partition between a more polar and an apolar solvent, like methanol water, partitioned against chloroform. You will be able to extract the lipids depending upon where they want to reside and which phase of that extraction. And then you can separate out the different lipids by using some type of chromatography technique. That can either be on a two-dimensional surface, like thin layer chromatography or TLC, or a column type technique. And then as you separate them, you can then analyze them either through gas chromatography, GC, or let's say a high-performance liquid chromatography. We call this HPLC. Separate those onto different peaks based upon the different strength of interaction of the different lipids with the stationary phases in these columns. You can see a variety of different peaks show up, and these can be discriminated from smaller lipids to larger ones will have different retention times. Different sites of saturation will also influence their retention time. And so you can quantitate the amount of the different fatty acids that are present, different lipids that are present in different tissues. So this is now leading to the science of lipidomics, right? So techniques that are used to characterize the concentration in different amounts of different protein structures. We call that proteomics. With regard to saccharide structures, we call that glycomics. And so now we have lipidomics as we characterize these biological lipids. Any questions about that? Okay, so the three types of membrane lipids then are phospholipids, glycolipids, and cholesterol. And let's begin to see what happens when we begin to organize these individual lipid components and ask them to self-assemble. I want to emphasize again that different types of tissue contain different membrane components. So there's going to be a different amount of protein versus lipid. There'll be different concentrations of phospholipids versus sterols. The types of sterols that are present are going to vary. And so you'll see these variations as you go from animal tissue to plant tissue. Most notably, plant tissue does not contain cholesterol. And also as you go from more complex organisms to single-celled organisms, you'll see that the extent of sterol in the membranes changes quite a bit, and the amount of protein component in the membranes for simple organisms increases quite a lot as well. So different organisms, different tissue types have different lipid components. Even different membranes within the cell also have different lipid compositions. And you'll see that cholesterol, for example, is really prevalent in the outer plasma membrane, but it's really not so much used to form the membranes that define the mitochondria. You'll find that sphingolipids are really prevalent in lysosomal membranes, but not so prevalent in mitochondrial membranes. So all of these different membranes form different functions and have different lipids included within them. Overall, then, the organization of the membrane looks like this. We'll talk about why we create this lipid bilayer in just a bit. But you can see that different lipids, different sterols, different protein compositions all will influence the different characters of these different membranes, and that we can also incorporate variation in terms of the different glycosylation sites, both on membrane proteins and also on membrane lipids as well. The integrity of that cell membrane is absolutely crucial if you want to keep the cell viable. And so many antimicrobials, antibiotics, their mechanism of action is to destroy the integrity of that cell membrane, to weaken it. Here's one example of an antibiotic called gramicidin, which organizes itself into these helical structures that creates a pore or a channel through that membrane and allows components such as cations to leak from one side of the cell membrane to the other. And we'll talk about pores, protein pores in membranes on Thursday when we talk more about membrane proteins. But this is one way in which you can conduct chemical warfare on a microorganism, use this molecule to poke a hole in it, and destroy its ability to control the concentration of cations within the cell. Here's another example of a similar molecule called gramicidin S, which also deforms cellular membranes. And this has been the focus of some research in my lab to try to come up with molecules that mimic this structure. And the critical things here is
very
that
bad
these
to
molecules, both the gramoside A and gramoside S, incorporate both these hydrophobic side chains and these cationic side chains as well. So these can be used to drive association with negatively charged phospholipid groups through these cationic side chains here, associate these molecules with the cell membrane, and then the hydrophobic groups bury themselves within the cell membrane, and these can be used to deform cell membranes and kill them. So here's some mimics of these gramicidin-like molecules that we've made, and this shows electron photomicrographs of cells that have been deformed using these molecules, and you can see that we've really poked some nice holes, and we've got good membrane destruction and good toxicity towards these highly resistant Staph aureus pathogens in these cases. This shows the structure of one of our molecules. The really interesting thing here is that we've been able to develop selectivity of these molecules for killing bacterial cells versus mammalian cells, and this is what you want in a drug. You don't want to just add some molecule that's going to destroy cellular membranes indiscriminately, because you'll be killing both your host human cells and the bacterial cells. What you really want to be able to do is get selective toxicity, and these molecules, very interestingly, have been able to do that, and it must be only because of the different lipid composition of bacterial versus mammalian cell membranes that allows us to observe this kind of selectivity.
I can fill in this.
All right, so let's talk about how lipids come together to form membranes. And we're going to go right back to some of the same kinds of concepts that we talked about when we were investigating why protein structures tend to form globular, compact, three-dimensional structures, driven by the segregation of hydrophobic surface area into the interior of those molecules. So if you consider an individual lipid group to be roughly wedge-shaped, and I'll encourage you to think about which of those lipid components that we've been talking about would have these characteristics, with a larger polar head group and a narrower hydrophobic tail. As you pat those hydrophobic groups against each other, because of the nature of the enlarged head group, they're going to form a spherical arrangement. So one way to pack these molecules in a very well-ordered way is to form these micelles, which is essentially just forming a sphere in which the polar head groups are all oriented towards the outside of the sphere. Now, as you continue to add lipid groups into these micelles and make them larger, as the aggregates become larger, you're still going to want to pack those hydrocarbon chains against each other. But there's two different ways to extend the size of these things. One is to break this from a spherical structure into a two-dimensional form in which polar head groups are presented on either face of the two-dimensional assembly. So this maintains a completely hydrophobic interior and polar surfaces on either side. The other is to create what's called
so it goes into the Krebs cycle to form NH.
And in this arrangement, we also have a bilayer in which a polar exterior is established, and we also have a polar interior surface which allows these liposomes to establish an aqueous cavity.
Yeah, the NTP synthesis is an electron. It's an electron chain, right?
Obviously, if you have a tendency to create curvature, you're going to want to create a tendency to form more of these liposome-like structures rather than these planar lipid bilayers. So I want to make an analogy now between our consideration of biomolecular structure. We talked about individual amino acids and how these can be brought together into larger structures in the form of protein polymers, and how those give rise to specific three-dimensional structures. We also talked about individual saccharide groups and how those can be organized. Again, we array them in either a linear or a branched polymer chain and look for ways that those can organize, for example, in the form of starch structures. In this case, when we want to take individual molecules, I'm going to encourage you again to think about the variations in the chemical composition of different lipid groups and then figure out what's going to happen as we begin to assemble them. But in this case, we're not going to assemble them by polymerization as we would for amino acids, saccharides, nucleotides. Instead, we're just going to allow these to come together through non-covalent self-assembly to form these organizations like micelles,
I think this is going to be a question.
Okay, so understand your essentially monomeric components, their different physical chemical properties, and what drives them to assemble. Now, some of these micelle-like structures, they should be kind of obvious to you because you encounter them all the time. And in fact, this is essentially what happens when you use soap in the form of detergent in your home. These are different types of detergent molecules. These look a lot like fatty acids, and in fact, fatty acids can be used in the form of detergents and soaps. A hydrophobic hydrocarbon tail and a polar, usually charged, but not always, head group. What happens when these things encounter a grease, oily structure or some dirt? Once again, they will orient themselves to sequester hydrophobic surface area. Very often, they'll do that by orienting their hydrocarbon chains around that grease or dirt particle, position their polar and charge groups on the exterior, and now this can easily be liberated from the surface simply by rinsing it with water. And some of these can also be used to associate with hydrophobic protein structures. And so this particular detergent right here, sodium iodesyl sulfate, we've already talked about in the context of protein gel electrophoresis. These molecules interact with proteins, denature them precisely through these sorts of interactions, orienting their hydrocarbon chains so that they associate with the more apolar units within the protein structure and denature them. So now we perhaps have a better appreciation for how individual lipid groups want to organize. We know that they can form micelles. Let's take a look at these bilayers as well. This is essentially the nature of what happens when we create a plasma membrane on the exterior of a cell. Right? We take these lipid components, we orient the polar and often charged head groups so that they're exposed towards either the extracellular or the intracellular aqueous environment, and we position those two different sides of the bilayer so that the hydrocarbon chains are sequestered in the interior. And right in the middle, you can see this area of perhaps decreased density. These are where the individual lipid chains are terminating from one side and the other. So you can simulate these things. Molecular dynamic simulations can be used to describe them. We can get a better understanding of the types of motions of these individual lipid chains. And what we see very often is that certain types of molecules are capable of diffusing through these membranes, but polar molecules are not. And that's because there's this huge region here, 30 angstroms, which is predominantly composed of exclusively hydrophobic groups. So this is a region in the membrane where a polar group just would not want to be. So we can take a look at, let's say, a water molecule, and the probability in one of these simulations of finding a water molecule either on the exterior or the interior of a lipid bilayer. And you'll see that water can be either on the outside or on the inside, but it really does not have a very good probability of being in the interior. Something like a glycerol group perhaps is not so bad, it can penetrate further in.
Why does
A
NADH produce more
CH2
protons
group that might be
and
present in
ATP
a more
than
hydrophobic
FAH?
small molecule,
know.
that might be
Don't know
more
if I'm
likely to
taking
be
gases
present in the
because
interior of
this
the
one
membrane.
has one hydrogen.
So this
or the-
defines really the hydrophobic character
drops the
of the
electron
interior of the
on
cell
the first
membrane,
protein
and the
instead
fact
of
that
the
these
second
things
protein.
are not freely
this because it's
permeable to polar small molecules.
This one.
So we're gonna have to figure out some other mechanism to get small molecules, including things like glucose, from one side of the cell to the other. Here are some common features of all biological membranes. They form these relatively two-dimensional sheet-like structures. They're anywhere from, let's say, 5 to 6 nanometers thick. We'll talk about the variation in thickness in a bit. They are composed of lipids and proteins. We've seen that saccharides can be used to decorate both of these groups. The membrane lipids are low molecular weight amphipathic molecules that form these bilayers in water. If membranes function, it's generally not because of the function of the individual lipid groups, but the functional characteristics are almost exclusively associated with the proteins that become embedded in the bilayers. And again, we'll talk more about membrane proteins on Thursday. The assembly of the lipids into membranes is driven by covalent interactions, and these things are asymmetric, right? So we've seen that there's two sides to the bilayer. The lipid composition on one side tends to be different than the other. Okay, and we'll talk about that right now. But first, any questions about that? Okay. So let's talk about the asymmetry of cellular membranes. So we talked about the different phospholipid groups that can be used for membranes, such as phosphatidylserine, phosphatidylinositol, for example. And you'll see that the portion of the bilayer that's exposed on the cytoplasmic side has a different phospholipid composition than the extracellular side. In particular, the extracellular leaflet is rich in phosphatidylcholine and sphingomyelin, whereas the intracellular side has got a more predominant composition of negatively charged phospholipids, such as phosphatidylinositol or 5-bisphosphate. So, asymmetry. Also, you'll see that the types of saccharides that are used to form glycolipids very often, like is shown here in this cerebroside, these saccharide groups are predominantly exposed on the extracellular portion of the cell membrane. So here's another schematic beginning to show you some of this variability, that the glycosylated regions of proteins and glycolipids are predominantly oriented on the extracellular side. And now you're beginning to get some kind of idea of how protein structures might become embedded or presented on the exterior of the plasma membrane. So, recognizing that both proteins and lipids can be extensively glycosylated, we can take a look at this photomicrograph of the exterior of a cell and realize that there is like a kind of a sweet coating all around the exterior of the cell. This is what's called the Biochem layer. And it's these interactions between saccharides, both present in glycoproteins and glycolipids, that really helps determine cell-cell interactions, will determine attachments and associations of cells to one another. So not surprisingly, when we look for aberrations in the way cells grow and organize themselves, lots of times these become manifest through altered cell surface composition, in particular, altered composition of sites of glycosylation. So here's a non-cancerous cell, and here's a cancerous cell. You'll see that the cancerous cell has got a lot of phosphatidylserine that somehow has made its way from the cytoplasmic side onto the extracellular side of that cell. And in addition to that variation in the asymmetry of the lipid components, you'll also see that cancerous cells have unusual sites of glycosylation on their surface. In particular, they have a much higher amount of sialic acid that's present in the extracellular region of those cancer cells. Okay, questions about that? All right. So far, we've really been thinking about cell membranes as being fairly static. And what I need to tell you about now is important models that have been established of the cell membrane that really emphasizes its more dynamic and more fluid characteristics. We already talked a little bit about fluidity with regard to, say, butter and olive oil. These same sorts of concepts can be applied to understanding the fluidity of cell membranes. And it's important to understand that any particular lipid group, for example, within a membrane is free to diffuse within that bilayer. So if we were able to label an individual lipid, let's say with a fluorescent group, and just watch where it goes over time, we would see that there would be two-dimensional diffusion of that lipid within one leaflet of the bilayer, but that in addition to that diffusion, there's also what's called transbilayer diffusion, in which a lipid can flip from one side to the other. These types of diffusions tend to be very rapid. This tends to be extremely slow, and if you just waited for it to happen spontaneously, it actually could take anywhere from hours to days. So now this kind of brings up a kind of an interesting issue with regard to the asymmetry of the lipid components within the cell membrane. Right? I told you that there's different lipids that are present on the extracellular side versus the cytoplasmic side. That must be managed by the cell. If it were just allowed to develop on its own, this type of diffusion would rapidly scramble all those components. And it also tells you that it must be catalyzed, because if we're trying to push one lipid from one side to the other, the cell's not going to want to take hours or days to wait for that to happen. Just like any other event within the cell, something needs to happen more quickly. The cell arranges some type of catalysis in the form of an enzyme to do that. And in fact, there are enzymes that associate with the cell membrane called flipases that speed this transbilayer diffusion from hours to a matter of seconds. So now we're beginning to see that there is motion of the lipids within these membranes. And we can watch what happens as we fuse two cells together. So this is one of the original experiments that was conducted to watch this happen. They took a human cell, decorated it with one color of fluorophore, a mouse cell, decorated it with, let's say, a red fluorophore, induced them to come together, and then just watched over time as the different colors managed to scramble themselves and diffuse all around that conjoined cell. So this told them right away that, yes, the membrane is not a static structure, that there is diffusion that takes place, and that there is some amount of fluidity here. That can be quantitated and evaluated through a technique called FRAP, or fluorescence recovery after photobleaching. This is a really cool experiment that also monitors diffusion of labeled lipids within the cell membrane. We're going to take a cell membrane, put fluorophores on there, and then we're going to zap a spot onto the exterior of that cell with a laser. It's a technique called photobleaching, and essentially switches off the fluorescence response of those dyes. It leaves a hole that's present in that cell membrane where there's no fluorescence due to that laser zapping. This is called photobleaching. And now we just see what happens to the fluorescence in that spot. Does it stay there or does it get filled in? And what happens is that there is lateral diffusion. The photo-bleached lipids migrate out. The fluorescently active lipids diffuse in, and that hole begins to fill in, and we can monitor the intensity of the fluorescence in that spot over time, and the speed at which we recover a fluorescent signal within this photo-bleached region gives us some information about the extent of fluidity within the cell membrane. This led two scientists, Singer and Nicholson, to come up with this model of the membrane. They called it the fluid mosaic model. And they described the fact that, yes, the membrane was composed of a bilayer, but that bilayer was present in a fluid state, and that the proteins were kind of swimming as components in this mosaic within the membrane, and that they were free to diffuse in those two dimensions. So it wasn't homogeneous in the fact that there were different components sitting in here, but that there was a lot of fluidity within this type of mosaic arrangement. So that led to this fluid mosaic model. We now have updated it. We still believe a large component of this fluid mosaic model, but we understand that it's not just free diffusion.
Yeah.
The different
Who's the second
components,
messenger?
especially the proteins, are not just wandering about willy-nilly. In many cases, they're kind of caged. So what's providing that caging? It turns out that there's cytoskeletal components within the cell that form, let's call them fences. So where there is proteins that are intrinsically associated with the membrane and are associated from one side to the other, they're kind of penned in by these cytoskeletal components and can be anchored into particular regions so that they're not free to diffuse completely throughout the cell surface. The presence of different proteins and the organization of the macromolecular components within the lipid bilayer, the plasma membrane, can be studied through these freeze fracture techniques. You can actually separate the two leaflets of the lipid bilayer and then use electron microscopy to study the organization of the proteins within either the inner or the outer surface. And what we've seen is that many of the proteins actually are organized so that they pass all the way through the cell membrane. And again, we'll get into a more detailed discussion of membrane proteins on Thursday, but this is just typical of one transmembrane protein that forms kind of a helical structure within the membrane in which hydrophobic amino acids are present within a single pass of the protein chain. A large portion of the protein then is within the cytoplasm, and a large portion of the protein chain is present on the extracellular region. Again, hydrophobic groups make sense that they would be sitting within the the membrane bilayer itself. And we already talked about the fact that these sites of glycosylation tend to be exposed on the extracellular side. Okay. Questions about that? We can begin to actually predict which proteins are going to be membrane associated by looking at the organization of hydrophobic amino acids along the linear polypeptide sequence. If we see a region that contains a large number of hydrophobic amino acids all together, this is giving us some kind of tip-off that's organized in this type of arrangement in which that section of the polypeptide chain is inserted within the cell membrane. So in addition to proteins that make a single pass through the membrane, this is another example of a membrane protein called bacteriorhodopsin. And this is a very typical architecture with 7 regions of the polypeptide chain making passes back and forth through the cell membrane. This is another example of a membrane protein structure in which multiple hydrophobic helices are arrayed and anchor that protein within the cell membrane. This is cytochrome BC1. All right. I think that'll be a great starting point for us to get into discussion of membrane proteins and how they can be arrayed with lipid rafts on the protein surface. We'll take a look on Thursday at the two main types of membrane proteins. The fact that many of them are inserted into the membrane. They're called integral membrane proteins.
That's a
Others of
secondary
them are
messenger.
associated with the membrane surface, and those are
Activated
called
phosphokinase.
peripheral membrane
Stimulates
proteins.
glycogen breakdown via activated glucose.
Here's the summary overall.
Okay, so this is a receptor.
Know the different types of lipids and the nomenclatures. Should be able to draw out a saturated or unsaturated fatty acid from the nomenclature, know where to put the sites of
And then
unsaturation. We
That's
talked
a
about
secondary messenger.
3 different types of membrane lipids, and you should understand
So
the
the
difference
receptor
between
protein
the
is
single
7
myelins and the phosphoglycerol.
Then the upstream
Understand
signal
the
protein will be the G
significance
protein.
of that,
GDP, right?
Yeah,
photo
GDP
leaching
and the
experiment
cyclase.
and how that can be used to
Oh, these are
understand the
both upstream.
Yeah.
features
And then the
of
second
then
cell
the cyclic
membrane,
AMP is a downstream.
and
No, it's a
understand how
second
that was
messenger.
used to
Oh, that's what I
establish
meant.
fluid
And then
mosaic
the
model
downstream
and
is
how
going to
that
be
model
one of
has
these
been
protein
fine-tuned.
kinases.
The last one.
And
The
then
phosphor-
on Thursday, we'll
phosphorylase
get into this issue
Yeah.
about
And
how
then
proteins can be
glycogen
organized on the cell surface and how they can be used